Good morning to everybody. It's Thursday morning, 10 o'clock, so means enjoy science. And of course, I have to thank you, the attendees. Without you, enjoy science make no sense. And in fact, today we have a very interesting talk from a very bright scientist. So I have to thank to Dr. Ciorulip for being with us, and of course to thanks to Emily Lang for all the work who makes this happening. To thanks to Dr. Giulio Draeta and Ignacio Vistuba for co-organizing this stuff, and to my friend and colleague Alessandra Ferraioli for co-hosting. And for today, I think it's a big pleasure for me to introduce Dr. Ciorulip. Why? Because it's all the time a pleasure to introduce young and bright scientists. She is trained in Hunan University, then in Georgia State University, and then she trained in the famous Rosenfeld Lab in UCSD. And in fact, all this very hardworking and very good training in the years is paying back, because the work that Dr. Ciorulip lab and combined with Dr. Liu Kyung Yang lab is absolutely brilliant work on one of the fields that I love the most, long non-coding RNAs. And in fact, it's a field with a lot of opportunities, fundamental science and therapeutics, and the work by Dr. Ciorulip is really proving clearly that long non-coding arrays are extremely important. I prize her work because you will see it's extremely detailed scientifically. It's all the time a pleasure to read the papers and to understand the thinking, and really I learn myself from the papers that she published. Of course, she has a lot of recognition in our institution. Last, I think a couple of years ago, she got the President Recognition for Faculty Excellence Award, which is a great recognition in our institution. Several from funding agencies. And with this, let me to invite Dr. Ciorulip to take the show, and good luck with the presentation, which, as you can see, it's a very interesting and very important for us, because it's not only cancer who kills the patient, but it's also what comes with the cancer in the same package. Thank you, Ciorulip, for being with us, and good luck with the presentation.
Good morning. Thank you so much for George and Alexandra's kind invitation for me to present in this seminar series. It's one of the major seminar series at MD Anderson, and actually I was trying to enjoy the time and then hopefully everybody can sit back and relax and enjoy my talk. So first of all, I want to echo with the theme of this seminar series, Enjoy Science. I also fully agree with George what he has been said, that I'm very happy because I'm doing all day what I like. Okay, I'm really happy to be here. And then today I'm majorly focusing on the genetic mouse model to show the fundamental functional importance of long non-coding in metabolic disorders. So Liu Qing, as George mentioned, is a co-PI of my project. So, and I have been focusing on long non-coding since we joined MD Anderson. So our long non-coding are different from microRNAs, so because they're long. So many small non-coding have been involved in the transcription regulation, and however, we're studying long non-coding in the signaling transduction. So frankly, long non-coding are very difficult to study. So one reason is, we know too little about them. So this is showing an example that this non-coding is called Link-A. Is about 1.5 kb. Is a relatively short one already. So about this non-coding is localized in cytosol. So far, we have identified some RNA motifs of this non-coding involved in binding with different proteins. You can see different areas of this non-coding seems mediate interaction with different proteins working as a scaffold. So obviously, you can imagine that the functional role of long non-coding could be very complicated. So as a brief summary, it has been found that long non-coding could serve as chaperones which bind to a kinase or metabolic enzymes. For example, many kinases or enzymes are regulated by their self-inhibitory domain. The binding of non-coding could modulate the potential conformational change of a kinase or enzyme, leading to activation or inhibition of the enzyme activity. The presence of non-coding could potentially serve an allosteric regulator to fine-tune the structure of catalytic pocket of a kinase or enzyme leading to the kinase activation. Non-coding could also associate with a kinase or replic unit to form a large RNA protein complex serving a hollow enzyme. So another scenario is that the binding of the non-coding to the effector of substrate may prevent recognition of the substrate kinase here. Of course, non-coding may also modulate the subcellular localization of any given kinase or enzyme. Another interesting thing is that we notice that many of those protein domains previously were not identified as RNA binding domains, they actually do. So you can see we're trying to list that a typical RNA binding domain, or unknown protein domains that may still functional to interact with RNAs. So we have been spending for the last 6-7 years to working on the genetic mouse models, trying to prove, why the non-coding are functionally important. So one of the very basic rationale we come up is, there's more than 50,000 non-coding in human genome. How do we find out which one is important, which one is not? So one of the strategy we're trying to use is we're trying to look at non-coding profiling for each of the major organs compared to embryo. So the reason behind is very simple. So if any of this non-coding is extremely highly expressed in the adult organ, it might serve very important function than the embryo. So using this strategy, we actually profile the non-coding expression in heart, liver, kidney, lung, brain, almost all the major organs of the mice have. In comparison with the embryo, we find the top list of non-coding, and then eventually it gets knocked up. Okay, so you can see we accumulating some mouse models. For example, this guy showing the homozygous showing extremely obesity of the mice, and then the next one you can see the animal of the homozygous actually they losing hairs. So and actually they also show growth retardation. Some animals is a little bit bloody, but this animal actually showing severe infection of the skin. So we think is involving the immunity of the local area. This animal shows growth retardation and the hyperpigmentation, which is a lighter coat of the animals. We're talking about this project slightly later. This one, the animal, showing the calcification of the muscle fibers throughout the body. Actually, it's very similar. The animal is dying. So we're going to show last two projects in today's presentation, and then we're also including some unpublished data. So one of the how non-coding actually when we do knockout, it shows a lighter color coat as I showed in previous slide. And actually it is this phenotype reminiscence of kind of phenotype similar to a human metabolic disorder is called phenylketonuria, shortly as PKU. PKU is a relatively common inherited metabolic disorder in newborns. Patients show light eye, light skin and hair color. The main issue of this patient is actually extremely high blood phenylalanine concentration. The reason is the enzyme, which is called PAH, which is the phenylalanine hydroxylase. This enzyme is the only one and a limited enzyme to catalyze phenylalanine to tyrosine downstream. For any problem of this enzyme, PAH, then the phenylalanine in the blood cannot be processed, leading to excessive accumulation of phenylalanine in the blood. And extremely high phenylalanine in the blood causing the brain damage. So the patient will have relatively shorter lifespan. They have mental retardation. For a 15-year-old children, they barely can do the addition or subtraction using numbers less than 20. So it is a big problem, healthy problem, and it's majorly a problem in Europe. And for the treatment of this type of disease is actually very difficult because this is a genetic disease. And then mostly the current treatment is basically phenylalanine restriction, which means all the proteins, including milk, meat, cheeses, nuts, all those things we intake daily, they are already rich in protein and phenylalanine, so they can be restricted. And the patient need to spend $1,000 to, particularly using those modified diet, which is totally phenolphthalein free. Okay, so therefore this is this disease so far, the current clinical trial and current treatment involving a kind of like phenolphthalein of pH enzyme enhancer, probably this only current treatment in addition to the phenolphthalein restriction. So from our side, we trying to knock out this non-coding called pair. Why it's called pair is because eventually we find that pair is actually associated with the protein pH. And then this showing a strategy to knock out this non-coding. So I want to mention that to knock out non-coding is hard. It's not as easy as protein target genes. You know, for the target coding genes, we introduce stop codon and that's it. And for the non-coding, we need to deplete the almost entire transcription body to make sure this gene will be removed. And then when we do the non-coded mice, the homozygous mice show the very significant growth retardation and extremely high of the blood phenolphthalein concentration. You can see by 4 to 6 weeks old, the animal already shows some phenolphthalein level about 1,500 to 2,000. So that's actually very similar to a typical PKU patient. And the animals also shows the lighter coat colors and very smelly of the cage. So our postdoc actually, she do not need to do genotyping. She open the cage and she can tell which one is homozygous. Okay, just by eyes. All right, and then these animals also show reduced lifespan, and they roughly can live about 1 year to 1 year and a half. A very interesting part is we observe this animal when they are 8 to 10 months old, they start to have seizures, and which is it's a phenotype we never seen before. So this is I'm going to share you some of the videos about how the animal behave as seizure. So seizure is actually a severe mental problem. You can see the animals kind of like freak out given at the time. And this is another example. The animal actually died after the seizure. So I'm going to share back, go back. Okay. Did I stop it? Okay, I come back now. So we start this seizure phenotype actually faithfully mimic the human patient, because for PKU patients, they will experience seizures when they're in the adult stage. Mechanistically, we're trying to identify why this non-coding actually have this phenotype. You see, the first thing is because we know too little about a non-coding. So the first thing is we're trying to identify the proteins that associate with non-coding. So this is using the mouse non-coding, and then to as a bite and look at what's protein associated with non-coding in the mouse liver, and we found PH, gene name PH4H, is a protein as a top candidate to associate with pair. And another question actually we have been facing in the non-coding field is, non-coding are rarely conserved between human and mice. So far the list is 35, which means that only 35 well-known non-coding are conserved between human and mice. So therefore, it greatly limits the research work about non-coding field. So we're trying to we think that although the primary sequence of the mouse non-coding is not aligned with human non-coding database, were that possible that PAH, the key protein associated with mouse, and human non-coding similarly in a similar way? So therefore, although the primary sequence are not highly conserved, they may functionally similar. So we're using the mouse liver or human liver, and using mouse pH or human pH to put on at the same time, and trying to identify the RNA binding protein, RNA binding with the mouse pH or the human pH on the right side. And then to summarize the data, it's interesting to see that a mouse, of course, we see pair. intact with PEH, and in the human genome, we identify a human noncoding now is called HULC, HULC. So this noncoding has been shown that to upregulate in liver cancer, which means it's highly expressed in liver tissue. It is also exclusively expressed in the human liver tissue. So we think HULC and a pair may all modulate or bind with PEH to modulate the PEH enzymatic activity. So this is to side-by-side compare the motifs coming from the CLIP assay to showing that they share the similar function, similar structure, secondary structure, that HULC and a pair may forming a stem loop secondary structure, which may involving the interaction with PEH. So the next question we're asking is how? And this, again, is another challenge. So this is actually based on the collaboration with Su Xing in experimental therapeutics. And his postdoc fellow, now his postdoc fellow is already a CIFRI scholar in Baylor College, one of 3 this year. So he actually helped us to indicate how HULC or pair associate with PEH in using relatively primary structure evidence. So he first tried to predict the 3-dimensional structure of RNA molecule, showing this stem loop structure, and how this stem loop associates with PEH. You can see on the right side, the green color is a PEH protein, and the left is the HULC RNA stem loop structure. They forming multiple interaction docking sites between the RNA and the protein. And when we zoom in, we find that the stem loop, especially the loop region of the RNA, associate with the regulatory domain of the PEH. You can see 2 amino acids play a very important role for this interaction. One is threonine 63, one is histine 64. Both amino acids has been identified to harboring mutation in the PKU patient. So if we mutate each of them or both of them, it will impair the interaction between the RNA and the proteins. So next, we're asking how this mouse RNAs. You can see on the right side in the purple part is showing the stem loop structure of the mouse RNA sequence. Yes, the primary sequence between mouse and human are different, but the 3-dimensional structure are quite similar. So when we try to align with the purple, which is the mice, and the blue is a human RNA, you can see they align each other very well. And of course, they also share the very similar binding pattern towards the regular domain of PEH. You can see they're even using the same amino acid and the nucleotide to mediate this interaction. So next, we are asking how noncoding may modulate interaction of PEH. So therefore, we're trying to use this in vitro enzymatic activity using either liver cells or liver tissues and then all the recombinant PEH. So we have done extensive experiments to verify the enzymatic activity of PEH is modulated by the noncoding RNA. In the left top, we're showing that this is an active control. On the right top, we're showing that the Y-type PEH protein is the enzymatic activity still could be enhanced if we incubate this protein with the HULC RNA molecule here. And in the bottom, we try to express 2 most established PEH mutants has been identified in PKU patients, and both of them show impaired enzymatic activity, which could be improved if we co-incubate them with the HULC RNA molecule. And on the right side, we're trying to summarize what we have been identified so far. You can see the presence of the HULC RNA molecule, it seems can improve the enzymatic activity of at least of the PKU mutant has been identified in PKU patient populations. And for some of them, they cannot. Mostly is because this mutation actually diminish the RNA protein interaction. So therefore, the presence of RNA cannot do anything. And then lastly, we're trying to demonstrate how can we improve the enzymatic activity based on our findings. So it is unlike with any other projects regarding to noncoding RNA. In many of our regular projects, once we're talking about this non-coding is involving cancer, right? So we're trying to knock it down using antisense oligonucleotide or ASO. However, for this project, because we are lacking this RNA, we're trying to restore back the presence or status of this RNA molecule. So therefore, we develop what's called RNA mimics. So in this model, we're showing that we developed this HOC RNA mimics representing the minimal sequence of HOC using synthesized RNA molecules. So the good thing of RNA mimics is we can synthesize any sequence. It's the modern technology allow us to do everything. Okay, so and then to enrich this liver-specific distribution of this RNA molecule, we link those RNAs to a GalNAc. This is the sugar. This multi-complex sugar structure is showing the specific binding affinity to the liver cell, which is hepatocyte. So therefore, it improves the liver distribution of RNA molecules. So you can see we're trying to treat the mice with those RNA mimics. So the interesting thing, firstly, is we noticed that once we gave the animals with the RNA mimics, for the first 24 hours, the effect is not very obvious. But after 24 hours, the effect RNA seems to show up. It's probably because the RNA mimics needs time to distribute to the liver and incorporate this endocytosis to incorporate inside of the liver cell. You can see we're using this, we developed this mouse model represent PHR408W mutant. Again, this is the most frequent mutation as been observed in PKU patients. And then when we try to treat them with these RNA mimics, after 24 hours, the animals start to showing reduced blood phenolane concentration. On the longer experiment, we're trying to give the animal with the relatively longer treatment. So we have been tested. The animal can tolerate every 3 days, but after 3 days, after 3 days, we have to continue giving them more and more RNA mimics to maintain a relatively low blood phenolane concentrations here. So in the United States, there is PKU actually still a big issue. So I'm not sure if any of you have babies. So you may know within 3 days of any infant being born in the hospital, they need to be blood test to measure the blood phenolane concentration in their using their foot blood drop. So this is a very standard clinical setting to screen for the PKU disease. So the proof of concept, we developed this RNA mimics. So composing this 20 nucleotide RNA molecule, hopefully we can improving for the application to the treatment of those PKU patients, in particular young babies. So you can see here. Okay. So one of the current clinical setting is to using a small molecule enhancer is called PAH enzyme enhancer called Kuvan. So this is already FDA approved. So currently in the United States, the most standard treatment strategy is using Kuvan plus low phenolane diet. You can see low protein diet, and then to reduce the blood phenolane of any PKU patient. So, but one of the major problem is many of those PKU patients, depends on which mutant they are harboring, they do not respond to Kuvan. For example, R4AW, which is the widely distributed PH mutant. So in our study, we're trying to find that our RNA mimics things can further sensitize the R4AW mice, which is PKU mice, to the treatment of Kuvan. So therefore, it may improve the outcome of this PKU patient. So currently, the RNA-focused therapies have been gradually widely distributed. So then on the right side of my slide, I'm showing the current knowledge about mRNA therapy. You can see for the biggest one is to insert the mRNA sequence through the liposome or AAV, and actually this is the most well-known RNA therapy is COVID-19 vaccine. So everybody, the vaccine shot you're taking is actually mRNA therapy. And then the second category is about ASO, like many non-coding RNA, if they are upregulated in cancer, the first strategy we're trying to think is antisense oligonucleotide, which is ASO. So those small RNA oligonucleotides will able to bind targets, the RNA targets, inducing those downregulation or degradation of those RNA molecules, allowing to show this anti-cancer effect. But in our case, we're trying to demonstrate this RNA mimics, which may restore the biological function of these RNAs, and then hopefully they can be widely involved in more metabolic disorders or inherited genetic disease. So you can see on the future directions on RNA mimics, they may involve in improving antiviral activity or increasing the anti-tumor activity as well. So this project is majorly handled by one talented postdoc fellow, Dr. Jian Li. Now she's already gone. She's going to the pharmaceutical company now for further development of the compound and treatment. So the next one I'm trying to move on is another disease called muscular dystrophy. So by the way, so far, are there any, do you have any questions? So please feel free to type in the chat box. I'll be happy to answer the questions or after my talk. So muscular dystrophy occurs about 1 every 10,000 children. So it is actually a very severe clinical problem. So the most frequent type of muscular dystrophy is called Duchenne's muscular dystrophy, or shortly as DMD. So this disease is mostly diagnosed with boys within their first 5 years. Most of them need a wheelchair by age of 12. And then their lifespan is generally around mid-20s. So it's a progressive disease affecting skeletal muscle, cardiac muscle, and smooth muscle. Once this disease affects the respiration muscles, then it is actually the final stage, and there's no way to turn back. So the loss and damage of skeletal muscles leads to muscle weakness and fibrosis. The damage of cardiac muscles leads to impairment of heart functions and eventually heart failure. The fibrosis of diaphragms results in respiration failure at later stage. So frankly, this disease don't have any treatment so far. So expect the physical support. You can see on the right side of this film showing that once with the DMD patient, mostly they need support from the back, and hopefully they have eventually a later stage they have the CPR or the other physical support. So therefore, this is the life of the DMD patient actually truly a tragedy. So when we're talking about the cause of this disease, is mostly caused by a mutation of a gene called dystrophin. So dystrophin is a major protein. It's actually the largest protein of human body. So this disease actually are caused by the mutation, truncation, alternative splicing, or insertion of these genes. However, currently, the mechanism about how can we restore the protein back, or what's the role with non-coding in the muscular dystrophy are totally lacking. And then the very first beginning is we noticed that this protein called dystrophin is harbor a zinc domain, so zinc finger domain. So zinc finger domain is a typical RNA binding domain. So therefore, we are asking, well, is there any RNA associated with this protein? So therefore, again, we performed CLIP assay using the protein antibody targeting dystrophin, and then trying to pull out the RNA complex and sequence of RNA. So the interesting thing is, for both human and mice, we observe H19 is the RNA associated with dystrophin. So here is we're working on a non-coding RNA which is conserved between human and mice. Okay, this is within the 35 non-coding alleles. So this is to validate interaction between H19 and dystrophin using the skeletal muscle and cardiac muscle. And on the right side is we're trying to do a CLIP rescue experiment, which is we try to knock down dystrophin or trying to knock out H19 and finding that if we knock out dystrophin, a DMD gene or H19 gene, the RNA protein complex are all diminished. So then we're trying to understand what is the functional role of H19 involving the dystrophin protein. So we noticed that dystrophin protein actually has been subject to strong ubiquitination. And here you can tell that this is a very strong ubiquitination ladder once we incubate E1, E2, and E3. So to summarize the long biochemistry experiment as short, we're trying to identify the ubiquitin system that mediates ubiquitination of dystrophin. You can see the ubiquitin molecule is first binding with E1, and E1 actually was passed by to E2, and then E2 eventually mediated by E3. So therefore, the subject will be added with multiple ubiquitination chain, and this chain has many different subtypes, but it's all out of scope of this talk. But once they have a large ubiquitination chain, the protein will go to the 26S proteasome for protein degradation. Well, probably it's easier to understand is if the protein is gone, then the protein is gone, the protein will not be there anymore. Okay. So on the top left, we're showing that the ubiquitination of dystrophin is very strong and then mediated by a E1, E2, and E3, and in the presence of the lysine case K48 of ubiquitin. On the right side, we're trying to determine the half-life of dystrophin. The dystrophin has a very long half-life. In average, it has about 55 days in vivo. And then we're showing that once we can knock out H19, this RNA, the half-life of dystrophin is greatly shortened. You can see the curve actually goes down quickly. So once we have H19 knockout, the half-life of dystrophin is about 7 days. So it's 60 shorter than the stable one. And then we identified that one of the cysteine, so you can see this cysteine is within the zinc finger domain of dystrophin. In zinc, again, just remind you, zinc finger domain is involving the interaction with H19. So this is the site actually modulates your ubiquitination of dystrophin. So once we introduce this mutant to the mice, the first couple, 12 weeks or 5, but the mice gradually show this progress, muscular dystrophy, and then they gradually show the fragile and weakness. And by about 45 weeks old, animals start to die. And this on the right side, we're showing that when you just open the animal, dissecting the animal, we see this throughout calcifications of those animals throughout their body. And you can see this is showing their median lifespan is about 46 weeks old. So one of the interesting things we're trying to prove is we notice that the dead animals, mostly they have food on their throat. So this is a very unique observation. We think is because they're smooth muscle and then they're on their chest, because they're so weak, they couldn't swallow. So therefore they may cause lung infection and then choking. Probably that's one of the reasons as animal dies. So I'm going to show you another 2 slides. So, okay, so then when we do the histopathological analysis about the animals, so this is actually, you can see we compare the animals in 4 weeks old, which is on the very left column, or on the 2 right column, which shows animals are 12 weeks old. You can see the animals, the muscle showing progress, muscular dystrophy. And you can see on the 4 weeks old, they start to already show some kind of damage on the contrant side. And then when we are 12 weeks old, the contrant side show more the muscle apoptosis and shrink. Regeneration is pretty big a difference. And then we show the diagram of the animals. When in 4 weeks old, the diagram seems okay, but if we're showing them in 12 weeks old, they are showing much thickness, thinner, and then more damage of the apoptosis of those muscle bundles. And then this is the method we're trying to measure the strength of the animal. So this is a very standard experiment. So we pull the animal two front paw, and then measure the strength, just like you're doing the pulling up or the fitness. And then you can see the animals, the strength is reduced. We're also using the bottom part showing the treadmill to test how long the animal can run. And you can see that actually we can test 5 animals at any given time. And on the right side is a statistic showing that once the animal are homozygous, they run slower and they run shorter, and they couldn't stand this extensive exercise. And I'm going to show you another video about how the animal looks like. So this is actually my postfellow. He actually developed this experiment. You can see it's not a typical equipment. He just built up. You can see he's using 5 cages to build a cave and trying to look at if the animal can hang on the very top of the cage wall. You can see this is a wild-type animal. He can hang on very well. And this is, on the right side, is a homozygous animal. You can see he put it up. Then now this homo animal just being holding. I can see he's barely hung on there. And then actually our postfellow was able to record how long the time that animal drops to the cage, and then to do the statistic analysis. Okay, I'm going back to my main slide. Okay, so let me see. I just want to make sure I go to my main slide. Okay. So next, we're looking to the heart of those animals. So for DMD patients, they usually will experience with heart problem when they're in their 20s, means later stage. So cardiac muscle were affected later than the skeletal muscle. And in our animals, we observe that animals start showing a heart function impact in about 24 weeks old. So on the right side, showing the histology analysis of those hearts, you can see the heart start to have the fibrosis, infiltration of those adipocytes, which is actually a very good sign of the DMD-associated heart disease. And then on the bottom side, it also showing the fibrosis of the heart muscles. On the left side, we're doing this electron microscopy, indicating that the heart muscle, particularly on the right side of the homologous mice, they show the broken of the muscle fiber and the sarcomere. Next, we're trying to look at the heart arrhythmia. Arrhythmia is difficult to test. You can see this is another challenge we actually have been facing. One of the biggest challenges of this arrhythmia is if mouse does not like humans. When we human do PKG, we stand there and spin still, but mouse don't. They'll move. And if I give them to put them to sleep, then the heart rate will be reduced, and then they won't reflect the original scene, what they have supposed to be. So therefore, we apply this system is we do a tiny implant. It's just like a tiny chicken bean. You can see this implant was inserted to the bottom part of the heart, have 2 probes, inserting one at the bottom and one at the top of the mouse heart, recording the mouse electrical activity of their heart. You can see in the bottom of the cage, there is a receiver. The receiver will transduce or pass all the signals to the computer. Eventually, we can recognize the mouse EKG signal. And on the right side is, on left side, we're showing that the mice actually have all different types of problems when they have this EKG recorded. You can see they have this sinus bradycardia and rest. They have AV block. The AV block means the part coming from the sinus node, the electricity coming from sinus node, but cannot be transduced while to the left and right ventricle. So therefore, the heart contraction was kind of impaired. You can see the EKG signal is way different from the healthy animals. And then the animal actually, once they show you this type of sign, those animals probably will be closing to the end of the lifespan. Okay, so next we're trying to develop a therapy. How can we improve them, right, for this dystrophy mouse disease? Again, we developed this RNA mimics, and then this RNA mimics improve against ubiquitination of dystrophin and improve the protein level of dystrophin in vivo. So therefore, it can reduce or slow down the progression of this muscular dystrophy disease. So this is to summarize that we have this RNA showing the interaction with the extended domain, and then it's the presence of RNA interfere with this ubiquitin system, which showing a TRIM63 and E2, therefore allows this dystrophin protein to be stabilized in vivo. So I'm going to skip this part because I think my time is short. Okay, so basically this is trying to, we're asking if we can, H19 is low in, which we can restore H19 protein in muscular dystrophy, but how about for healthy animals, for wild-type animals? And then again, we're using this RNA oligonucleotide, and then now this time we're trying to treat with healthy animals that don't have this muscular dystrophy disease. What we can do? So it's actually induce this muscle hypertrophy. So which means the animal, you can see this is the image mapping of the bone, lean, or fat map of the animals. You can see the animals, if they have long-term treatment, it's more than 6 weeks, 6 months treatment, the animal is actually showing stronger of the muscle. They have more muscle, they have less fat, and then they have things are more energetic. And then we also challenge with these animals with the high-fat diet to see what's going on, right? Because the amount of skeletal muscle is actually closely related to obesity. So we challenge with those animals with high-fat diet and then treat those animals with those small molecule RNA mimics, and you can see the body weight of those supposedly fat mice are significantly reduced. We also see improved fatty liver and reduced fat tissues of those animals. To brief summary, so this project was handled by two postdoc fellows, Dr. Yahua and Yazhen. Yahua has gone back to being an independent investigator now. Now he has his own lab. He showed me he has big lab and he has his own startups. He has been having his own lab and his own projects now. So in a brief summary, we're showing this Noncoding modulates the ubiquitination of dystrophin, and then if we introduce DMD mutant to the mice, the mice exhibit muscular dystrophy and cardiomyopathy, and then H19 RNA mimics improve or slow down the muscular dystrophy of this mice. If we use H19 gain-of-function mimics, it may promote muscle hypertrophy. So I think I still have 5 more minutes. I'm going to show you some of the newest data we have. It haven't been released to anybody. This is my first time to present this project. I believe George will be very happy to see that. So again, this is we're asking which noncoding is extremely highly expressed in heart compared to embryo, and one of the top candidate we call is C10. So this time is the slightly different is we did this tissue specific knockout. So on the left top, we can see is we insert 2 LOXP cross this gene transcription body, and then once this homologous cross with the particular cree is allowed for the tissue specific knockout of this noncoding. And the first time we use is heart specific cree MYH6. So to be honest, this animal, the triple positive animal, just come out last year, fall of last year. But we're very excited to see that animal shows sudden death. The animal generally shows sudden death between age of 8 weeks to 14 weeks. The lifespan, we're still analyzing lifespan of those animals, but so far we haven't observed any animal survive longer than 14 weeks old. And on the right top, we're showing how the heart looks like. You can see on the right side, we see the heart are enlarged. And then they actually particularly have big right area of on the right side. So this is this project actually is handled by my research technician. So I introduced him slightly later in the last slide. And when we do MRI, it's showing that compared to heterozygous, the homozygous animals showing enlarged RV right ventricle, enlarged RV left ventricle as well. This is about 12 weeks old of the animals in both diastolic and systolic conditions. And then if we move this MRI section to the top, on the right part, I'm trying to show the MRI image between on the top of the ventricle. So therefore you can see both left and you can see the aorta as well. And you can see for the aorta, which is the main aorta, you can see it's enlarged, and also the RA is also significantly enlarged in homozygous compared to the heterozygous. And then this is the echo imaging. Oh, let me show you this. So actually, my research technician, actually, he prepared the video for the, here, am I sharing this? So I'm trying to show you the video about the wild type on the left, heterozygous on the middle, and the homozygous on the right. All of them are 6 weeks old. You can see the wild type animals shown pretty good. This is how normal echo look like. The heart beating properly, and then the heart have strong contractions. And again, this is heterozygous. Heterozygous already show impaired heart contraction. You can see the heart is weaker. They kind of like shaking instead of strong contraction. And on the right side is the homozygous. You can see the heart is even worse. They barely have any functional contraction to pump the blood to the aorta. Okay, so let me come back to next slide. So now on the right side is the statistical analysis. So basically, we look into the thickness of the heart wall. We look into the volume of the left ventricle. We also look into the ejection fraction, which is the golden standard parameter to indicate heart failure. And actually, you can see by 6 weeks old, some animal already show heart failure, which is the ejection factor lower than 30%. And then so this model is pretty different from any of the previous studies. So you know, see all the coding genes multiple coding genes has been shown. For example, Notch has been shown to involve in heart development. But all those animal models actually shown either embryonic lethal or fatal right after the birth. In our model, the animal can survive till adult age, probably between 8 weeks old to 14 weeks old. We don't know what's the difference. We're still working on the mechanism. And then on this side, please forgive me, on this figure, this is sectioning wasn't done properly. Actually, Andrew, he's the research technician. He's handling solely on this project. And then this is actually done by him. So it's not the perfect one. So we are still seeking to the technical the experts to give the very good representation about the imaging. But here showing actually this is the 8 weeks animals. You can see the heart is pretty large compared to other stories. And then on the top part, we're trying to show the right mitral valve, which is here. And then the separation between the atrial and the ventricle. And then on the bottom part, this is homozygous. So interesting thing is the left and the right atrial things are connected. You can see from here, it looks like there's no separation between left and right. And then on the bottom part, if you zoom in, the left and the right ventricle seems also lacking the separation here. And both of them actually are most frequent cause of congenital heart disease. So if you go to the hospital, for the children 3 to 5 years old, they mostly happens is congenital heart disease. They have this problem of ESC or VSC, and most of them they need to be repaired by surgery. So this is in our case is we think the narcotics noncoding seems particularly involved the valve development and this statistical defect. So like I mentioned, this project is solely handled by my research technician. So he just graduated from master degree. He joined the lab for about 6 months. So when he just joined in, his position is like, okay, can you handle genetic mice? He's going to handle genetic mice, right? But when I talked to him about this mouse, we observed the mouse sudden death. I'm asking, do you think you want to try the imaging MRI or echo? He said, I do not know how, but I can learn. So he learned everything. He learned from our animal facilities. He learned from our collaborators in all other nearby. So within 6 months, he collects very strong preliminary data. I'm very proud of him, and hopefully he can be an MD student or PhD student in shortly. So therefore, he is doing what he's having. So like I mentioned, I think this is the theme of our seminar series, and we want to be enjoy the science. We want to enjoy our projects and just look into what's going on. And as long as we can learn about it, we will be something fun will be fine. And then lastly, I'm going to thank you our research teams. Okay, I need to say big thank you to Dr. Yang. Dr. Liu Qingyang, he is co-PI of all the projects. He is controlling the big shape of our direction of our lab, which direction, which candidate we go through all together. So this is without him, now this project will be here. So our research teams, so like mentioned, Yajana already goes to the pharmaceutical companies, and then Yaohua already go back, and then we have some newcomers, and then Lisa is our graduate student, and then Andy is our research technician handling our new project. And thank you for our collaborators. So I believe for all the trainees, if I have the take-home message today is, I hope you guys can enjoy science and be open-minded. Whatever we have, just look into. It will be great to see that your observation is totally opposite from our initial hypothesis, which is good. That means that something really, really interesting could be fine. And then I hope that we all be happy, all enjoy your stay in MD Anderson. Okay, thank you so much for your attention, and I'm happy to take any questions.
Thank you very much, Rose. This was a wonderful talk. It's in fact excellent science and show how large is the spectrum of involvement of non-coding RNAs. And let's start on the question. So Swati Mohapatra, a straightforward question. Did you see any change in the creatine kinase level in the peripheral blood of any of these mouse models that you presented? Mostly deep muscular dystrophy, I suppose.
I see. For muscular dystrophy, the H19, we think H19 is pretty large. It is about 2.7 kb. So we think H19 is within the skeletal muscle bundle. So therefore, it's not, the functional mechanism is not in the blood. It's within the tissue. And then we do observe that CK levels high is very standard. All those are showing the injury or damage of skeletal muscles. And then we also observe that for the dystrophin associated with dystrophin-associated complex is called, there's a multiple big protein complex. If we remove H19, this complex is also got impaired.
Okay, thank you. Now another question from Jit Kong. She is from Singapore. So very large attendance. Excellent talk. I probably missed the information during your talk. Is C10, her specific long non-coding RNA, one of the 35 conserved long non-coding RNAs? If yes, have known loss of function mutation of C10 been found in patients with her failure?
Okay, good question. So C10 is now within this list of 35, but it does have a high reservation. So we have a way to bioinformatically calculate how conservation between mouse and human non-coding RNAs. So our list is more than 35. We have roughly around 300 non-coding RNA lists, which we think are showing kind of conservation. So this non-coding RNA actually does have a homologue in human conserved within the gene loci. So you can see the whole loci gene structure, RNA direction, and nearby genes are all preserved. And then the human homolog is also very strictly highly expressed in heart and skeletal muscle. Also, we have did some bioinformatics analysis. So this human non-coding, not mouse one, the human non-coding is downregulate in part of the disease involving the ischemic heart disease and certain type of cardiovascular disease. So we're still trying to validate the human relevance of the human homolog and this non-coding, but it seems current data seems promising. We also need collaborations, internationally to hopefully we can detect something interesting. Thank you for the question.
Thank you. Now from Dr. Jan Parker Torborg, who is a master of the mouse facility in our place, and we thank her for all the work. One question: for the HER-specific long non-coding RNA studies, did you see fibrotic plaques in the atria and ventricles? Do you see problem with the pulmonary arteria?
Okay, that's a good question. So, we are still kind of like behind to characterize the physiological of the heart. So you can see I'm desperately need a postdoc fellow working on heart. So far is we do observe that the heart muscle, means the ventricle and the valve, both have problems. So my suspicion is, it's actually very early stage of this project. My suspicion is this non-coding RNA do knockout. It affects valve development at the early stage, but it also affects the muscle fiber at the later stage. So actually, we are crossing this angle with inducible knockout, which is alpha HMC mice. Hopefully, we can have in the later stage of the mice, so we can observe what is solely only affected with the muscle fiber and not the valve. So therefore, we can separate both the phenotypes. Yes.
Thank you. Now, Sanjay Singh, Dr. Lin, really enjoy your presentation. We know that post-transcriptional modification play a key role in formation, stabilization of secondary, tertiary structures of RNAs. What is known about post-transcriptional modification of long non-coding RNAs? H19 locus is known to be maternally imprinted. What may be the consequences of imprinting with respect to muscular dystrophy? Thank you.
Okay, that's a good question. So the post-transcriptional modification of RNA has been, many groups study this, including, I believe the most frequent one is RNA methylation, C6 methylation. So our lab, we don't touch this part. So to be honest, we have no idea about any of our non-coding RNA candidates, whether they have modified or not. We didn't try this direction. So for H19, it is a printing gene, and it is widely expressed in embryo, but when in adult stage, H19 is solely expressed in skeletal muscle. So, and actually we did the RNA scope to indicate in heart muscle it also have some kind of adequate expression of H19. So I believe, in the adult stage, H19 only expressed in heart and skeletal muscle and involving the muscular dystrophy. Thank you.
Thank you very much. And one more question. So, what do you think about, you show these very nice long non-coding RNA mimics, which are fragments of the gene. So what do you think about their use in cancer cells and for cancer patient therapeutics?
Absolutely. So generally speaking, current cancer study, we're only focusing on the upregulated non-coding in cancer. So if we're focusing on non-coding are downregulated in tumor compared to normal, then those non-coding may serve a tumor suppressor role. Under this scenario, we can think about to restore the expression of those RNA using RNA mimics. I believe so far this part is right behind. Even in our lab, we haven't keep seriously looking to the RNA candidates that are downregulating cancer. That's a very good point. Give us good directions.
So with this, thank you very much, Chunru. It was a wonderful presentation. In fact, a wonderful example how to enjoy science. And for all the one who attend us, enjoy science, have fun, and good luck with everything. And see you in 2 weeks for another excellent talk. Thank you again from Dr. Tira Cascone. Thank you again, Chunru, and good luck with everything.